Good evening and welcome to Monobiology. This evening we're going to talk about the errors that may occur during meiosis. These errors are called non-disjunction. Essentially, this occurs when the spindle fibers do not attach properly to the centromeres, and the result is either the homologous chromosomes or the sister chromatids do not separate. As you can see in the picture here, we have our chromosome, and in the center of the chromosome, or somewhere thereabouts, we have our centromere. And here we have a spindle fiber, and the spindle fiber is not attached properly to the centromeres. Remember that the spindle fibers are what coil and bring the chromosomes, or the chromatids, towards the opposite poles of the cell. If they don't attach properly, they won't be able to do that. They won't be able to pull the chromosomes from the middle of the cell towards the pole of the cell, or the side of the cell, or the top or the bottom of the cell, depending on how you want to look at it. Why is this a problem? Why is it a problem that the chromosome or the sister chromatids don't go exactly where we want them to go during the process of meiosis? Well, recall, the process of meiosis is designed to create gametes. These gametes are supposed to have the haploid number of chromosomes. If the chromosomes don't move to opposite poles like they're supposed to, or the chromatids during meiosis II, then the correct number, or the haploid number of chromosomes, will not result in each of the gametes that are produced. So as we can see here, there are actually two possible scenarios, or two possible times, when non-disjunction can occur. In this situation, here, non-disjunction is occurring in meiosis I. You can see in this diagram here that the homologous chromosomes are supposed to be separating, but because the spindle fiber didn't attach properly to this centromere here, both of these homologous chromosomes are moving to the left side. And at the end of meiosis I, notice that instead of having 2 chromosomes in each cell, which would be the haploid number, we actually have 3 chromosomes in one cell and 1 chromosome in another. Now they undergo normal meiosis II, and we end up with this. Right? The same thing can be said here, except for in this case, it happens during meiosis II. Right? Again, the result is either too many or too few chromosomes in each of the gametes. If a normal sperm cell fertilizes a n+1 or haploid +1 egg cell, the result would be a 2n+1 zygote. Now 2n+1, an example in real life of a 2n+1 would be Down syndrome, where it's trisomy 21. So there would end up being 3 chromosome number 21s. That would be an example of this. An example in real life of an n-1 individual, or a 2n-1 individual, would be Turner syndrome, where there's only one sex chromosome. It's called monosomy. So, as stated earlier, there are two situations, or two times, when non-disjunction can occur. The first one is during meiosis I. The picture that you see here is the same picture that was drawn on the board during class. So error type 1 is during meiosis I. Notice I've labeled each of the phases so that you can kind of follow the process. Right? It's a little bit more in detail than the pictures I showed you a few minutes ago. So metaphase 1 is where the spindle fibers do not connect properly to the centromeres of the chromosomes. Okay, so in this particular situation, the big blue chromosome is the one that is not connecting properly, or the spindle fiber is not connecting properly to it. Right? So That technically isn't non-disjunction. Non-disjunction is here in anaphase 1, where when the chromosomes are being pulled to opposite poles, see how this chromosome isn't being pulled? It's kind of hanging out in the middle. That is non-disjunction. Okay, so the first mistake, I guess, if you want to call it that, occurs during metaphase 1, in this case, where the spindle fiber did not attach. The non-disjunction doesn't actually occur until the spindle fibers start to contract and the chromosomes are being pulled towards opposite poles. As you can see here, this chromosome, the big blue one, is not being pulled, and therefore it's called non-disjunction. What's the result of this? Well, if we fast forward to telophase 1, what we see is that in this haploid cell, we have the N-1 situation. We only have one chromosome. We started off with 4, so the haploid number should be 2. Here we only have 1, but notice down here we have the N+1 situation. So neither of the cells at the end of meiosis 1 actually have the correct number of chromosomes. This is bad. This means that every single gamete produced from this one process is going to be incorrect and potentially lead to some sort of disorder in the resulting zygote after fertilization has occurred. Now let's look at what happens for non-disjunction during meiosis 2. Right, so we're going to follow both of the cells. We're going to assume in this case that we're in meiosis 2, but we're going to assume that the meiosis 1 that occurred was normal. So no non-disjunction occurred during meiosis 1. So we're going to look at this, and here we see the non-disjunction now is occurring in the little blue chromosome, except, sorry, this technically isn't the non-disjunction, but this is where it's going to occur. Right now, all we see is that the spindle fiber is not connecting properly to the, we'll call it the bottom little blue sister chromatid. Okay, so if we follow that through, this one's normal, all the way through. Notice there's 2 chromosomes here, normal. They're separating normally, and you end up with the normal number of haploid. So let's follow this one through. Anaphase 2. Notice that the sister chromatids for the big blue chromosome pulled apart, no problem. Here, because the spindle fiber didn't attach properly in metaphase 2, this entire chromosome is pulled towards the top pole. That is the non-disjunction. The result of that, if we fast forward to telophase 2, is that one of the cells will be N+1, and the other one will be N-1. See, there's 3 here and only 1 here. So in this situation, if these, let's say they were sperm, if these were to fertilize an egg, this one would result in a trisomy, and this one would result in a monosomy. Okay, so the potential there is to lead to some sort of disorder if they happen to fertilize or be fertilized if they're egg cells. Now, if you look at this really closely, notice that only 2 of the 4 gametes are affected by the non-disjunction. Right? You have this one and this one. If you think about the previous one, you will have noted that all 4 of the gametes were affected by the non-disjunction. So the error during meiosis 1 is much more potentially dangerous than the error during meiosis 2. Having said that, because the non-disjunction is definite here, in other words, because the chromosome definitely gets pulled entirely to one side or the other, this is actually more likely to occur. So the other one's more dangerous, but this one's more likely. And that, in a nutshell, is non-disjunction.